Our next speaker is our very own Emilee Knowlton. So Dr. Knowlton gained her PhD in infectious diseases and microbiology from the University of Pittsburgh under the direction of Professor Charles Ronaldo, identifying immune responses to lytic infection with human herpes virus 8. She joined ProImmune back in 2013 after completing her postdoc in Professor Ronaldo's lab, and she works on the ProImmune team providing innovative solutions for our clients that radically improve our understanding of both desired and unwanted immune responses. And Emilee today is going to be talking by way of a bit more introduction to the following speakers as well about our Pro5 MHC class 1 Pentamers and ProT2 MHC class 2 Tetramers for tracking antigen-specific C cells. So over to you. Thank you, Emilee.
Yeah, great. Thanks, Jeremy, for that introduction. I think this will fit quite nicely with what we've already heard from some of our other speakers, and then, as you mentioned, serve as a nice introduction for the ones to follow. So characterizing antigen specific T cells is really a crucial step in confirming functional T cell epitopes and interrogating T cell populations in preclinical research. Antigen-specific T cells can be measured all through the immune monitoring phases of clinical programs to help evaluate the efficacy of your vaccine or therapeutic in the context of a wanted immune response, as well as monitoring unwanted T cell responses in patients who have been dosed with something like a biotherapeutic and are at risk of anti-drug antibody formation. So success in all of these programs are dependent on having reliable reagents to track antigen-specific responses that perform consistently regardless of the disease or the antigen. So today I'll be sharing our Pro5 Pentamer and ProT2 Tetramer technology to track those antigen-specific responses. And of course, we're all joining with different backgrounds and disciplines, so I do have just a brief slide to introduce the principles behind the technology. Shown here are the antigen processing pathways. On the left is class 1, which is the processing of intracellular material like a virus or tumor antigen into smaller epitopes, typically 8 to 11 amino acids in length. And those are presented on a class 1 molecule to the T cell receptor on a CD8 T cell, which activates our cytotoxic immune response. On the right, we have processing of extracellular material into longer peptides, generally 15 amino acids or greater. And those are presented on a class 2 molecule like HLA-DR, DP, or DQ to the T cell receptor on a CD4 T cell, which initiates the helper T cell immune response. So the multimers that I'll be describing can help to enumerate these T cell populations by binding to the T cell receptor. So up first are our class 1 pentamers, and you see the pentameric structure shown here, with 5 MHC peptide complexes that bind to that T cell receptor, and then 5 fluorescent tags, which allow for very bright and uniform staining. And this is configured through a coiled coil domain, which is unique to the pentamers. So you'll hear about a lot of different applications today over the next several slides, but these include things like separation of antigen-specific CD8 T cells, phenotyping by flow cytometry in combination with intracellular cytokine staining, tissue staining, cell sorting, and even looking at multiple HLA restrictions or multiple antigen specificities within the same sample. And what speaks to the versatility of these reagents are the number of scientific publications. With more than 1,800, the pentamers are the most referenced class 1 reagent on the market. So because of the unique configuration of the pentamers, we have a very flexible labeling option. We can provide them pre-labeled with either APC, PE, or a biotin tag, or you can get them unlabeled plus fluoro tag. And those unlabeled pentamers have an extended shelf life of up to a full year, and using them doesn't add any additional time to your staining protocol. You'd simply add the fluoro tag, which binds to a high affinity epitope tag on the pentamer. The same time you would add your anti-CD8 antibody. So this is a really nice option for groups who are going to be performing studies over a longer period of time, like a clinical trial, because you can get a lot of pentamer from a single batch, QC them, aliquot them, freeze them down, and then use them as needed over the duration of the study, which helps to limit that batch-to-batch variation. We have over 700 catalog items spanning viruses, cancer, autoimmunity. A lot of those are available in 1 to 2 days or as long as 1 to 2 weeks. And if we don't have your specific epitope of interest, then we can generate a custom pentamer for you at no additional cost. And this is quite a simple and straightforward process. You provide us with the allele restriction and the peptide sequence, and we use an internal CIF-PyThi program to evaluate those anchor residues. And if we accept the sequence, then you'll have your reagent in about 6 weeks' time. So our first case study comes from Professor Tao Dong at the University of Oxford, where they were looking at T cell responses in convalescent COVID-19 patients. And they had access to individuals who had recovered from both a mild form of the disease, as well as 14 patients who had recovered from severe COVID-19. And they were interrogating a panel of 6 different pentamers with different HLA restrictions of epitopes from SARS-CoV-2. And what's shown here is the representative data for an HLA-B4001 restricted NP sequence. So you can see their gating strategy, as well as the CD8 versus pentamer stain with a very nice, clear SARS-CoV-2 specific population. They did also include different surface markers for phenotyping, including CCR7, CD45RA, CD28, and CD27, and concluded that there was a higher magnitude and a greater breadth of memory response in the patients who had experienced the severe form of the disease. And those pentamer positive or SARS-CoV-2 specific T cells did have a central and effector memory phenotype. So you can hear this talk from Professor Tao Dong on our website, where she gave a talk at a recent previous conference. So this next case study comes from Barbara Serafini's lab, and this is a really great example of some of the various applications for the pentamers. Their focus is on the role of EBV-specific T cells in the pathogenesis of multiple sclerosis. And these images actually come from 2 different manuscripts, the first from 2019, and then the second one was just published last month. So in the first paper, they confirmed the presence of EBV-specific CD8 T cells in the brain lesions of MS patients using immunohistochemistry in the pentamers. And they chose 2 B0801 restricted EBV sequences, the EBNA3A and BZLF1. And they found that these EBV-specific T cells were at a higher frequency in the brain than influenza-specific T cells, in which case they also used influenza pentamer. And that staining was specific because when they used an allele mismatched control, they didn't see any staining. Those EBV-specific T cells were recruited to the brain and interacted locally with the EBV-infected B cells, which is likely to contribute to that MS pathology. In the more recent paper, they used the same EBV pentamer specificities, as well as stained for PD1, which is shown in this immunofluorescent staining below. On the left, you have PD1 staining. They used a pooled approach for the pentamers, and then you see them merge in the final column. And virtually all of the EBV-specific CD8s did also express PD1, suggesting they used this as an immune evasion strategy for those CD8s to persist and contribute to the pathogenesis. So this is a really nice study from Dan Baruch and Tom Henley's group using both the class 1 pentamers as well as the class 2 tetramers to generate a viral antigen-specific T cell therapy. So they combined biotinylated multimers onto streptavidin-coated beads in combination with an anti-CD28 antibody to generate micro-artificial APCs, or what they called Viper particles. And as a proof of concept, they first started with CMV immunodominant epitopes to determine the ability of those Viper particles to stimulate the TCR and expand the T cells. So they chose CMV PP65, which is an AO2, a 1 restricted sequence, and then CMV GB, which is a DRB107 restricted sequence. And their methods are shown up here in figure A. They isolated T cells from their patient, exposed those T cells to the Viper particles for 7 days to allow for T cell expansion, and then looked at those T cell populations by flow cytometry, which is shown in part B. So you have the PP65 pentamer versus CD8 and the GB tetramer versus CD4. So in comparing day 0 to day 7, you see a very nice expansion of the CMV-specific CD8 and CD4 T cells. And this was significant for the three donors that they explored. They then transitioned from CMV to SARS-CoV-2 as they wanted to explore this as a potential therapy for COVID-19. So rather than using the CMV pentamers and tetramers, they used a SARS-CoV-2 spike-specific pentamer, this YLQ sequence, which is restricted to AO201. And they used those Viper particles to expand the T cells, which is shown here. So again, a very nice, clear SARS-CoV-2 specific expansion. They also did intracellular cytokine staining for interferon gamma, TNF alpha, and IL-2, and found that those pentamer positive cells were much more polyfunctional, which are shown here in this dark part of the pie, meaning they made all three of those cytokines. So this represents a novel platform for cellular therapy, which could be applied to COVID-19 and potential future pandemics. So we've just heard a little bit about our class 2 tetramers, but just like the pentamers, we have an expansive catalog with more than 200 specificities, including viruses, autoimmunities, allergies. We also have tetramers against different biotherapeutics. But just like the pentamers, if we don't have your epitope of interest, then we can do a custom synthesis at no additional cost for any of the alleles that you see here for human or mouse. These are pre-labeled with either APC or PE, or you can also get biotinylated monomers. So this next study comes from Peter Deek's lab at Drexel, and his focus is on the tolerogenic dendritic cells for a potential therapeutic in autoimmune disease and transplant rejection. And a tolerogenic dendritic cell can induce a naive T cell into an antigen-specific Treg. So they've developed a push-pull immunomodulation practice, or PPI, to induce these tolerogenic dendritic cells using toll-like receptors and immunomodulatory small molecules. So throughout the paper, they explored 40,000 different PPI combinations using different TLRs and small molecules, and then looked at the antigen-specific T cell responses on their top PPI candidates. So to evaluate those T cell responses, they used the model antigen of albumin in vaccinated mice, and they isolated their T cells and bone marrow-derived dendritic cells. Those DCs were treated with the different PPI formulations, then co-cultured with the T cells, and then they could look at those responses by flow, which is shown here in part A. So they did find an increase in the antigen-specific Treg population using an ovis-specific tetramer for their formulations PPI-7 and PPI-9. Here are those populations here compared to the naive cells. And this also resulted in a decrease of antigen-specific CD8 T cells, in which they used the synfecal class 1 pentamer, which is shown here. And that was also demonstrated in part C, where you see a larger ratio of the antigen-specific Treg cells to the effector CD8s for the PPI-7 and 9 formulations. So these represent potential ways to generate robust tolerogenic dendritic cells and then strong antigen-specific Tregs. Okay, so this last case study comes from Jennifer Juneau's lab, and this is another really nice talk that can be found on our website that she presented from a previous conference. And here they are looking at CD4 T cell responses in convalescent COVID-19 patients, as well as patients who had received a spike-encoding vaccine. So this is the data for their spike-specific epitope that's restricted to HLA-DRB1501. And on figure A, we have a convalescent COVID-19 patient who expresses DRB1501. And you see a really nice, clear tetramer-positive population, which was not seen in an individual that did not express that particular allele. In the vaccinated individuals, again, following vaccination, you see a nice increase in the SARS-CoV-2 specific CD4s, which again was not seen in the negative control in that individual without DRB1501. They did also include some different phenotypic markers and showed that those tetramer-positive cells were circulating T follicular helpers and memory CD4s, which of course can contribute to the long-term protection against further exposure to the spike protein. Okay, so successful immune monitoring programs really require detailed and efficient characterization of antigen-specific epitopes. And I'm showing you several examples of where it's really important to have a very reliable and consistent tool to track those antigen-specific CD8s and CD4s, which anyone at ProImmune would be happy to talk to you about. So our information is listed right here. And then over the next couple of months, if you're going to be attending any of these conferences, we will be there. We'll be all around the world. So please stop by and say hi to us. We'd love to see you. Thank you.
Great, thank you very much, Emilee. So we have a question in the chat from Guwai Wu. Says, can micro APCs activate naive T cells without co-stimulatory signal, or can it just activate memory T cells?
That's a good question. I think they did actually include some co-stimulatory signals in that paper, which is they just included some additional cytokines. So that's a good question. That might be a good one for the author. They might have looked at that as well.
Yeah, thanks. So yeah,
Mhm.
so actually one thing just in relation to it just sort of triggers a thought in regards to looking at sort of antigen-specific populations with multimers. I guess one thing just to be aware of is when you are stimulating the T cells, it's important to ensure that the T cell receptors are still expressed. So actually when you do stimulate cells, for example, with peptides or other approaches, you do need to typically allow some sort of recovery of the TCR to kind of gain re-expression, so that you can then actually have some protein for the MHC peptide complexes to actually bind to. That's just something to be aware of as more of like a technical hint. One other thing that we often get asked, which may be pertinent for the audience, is thinking about the use of DNA barcoding in the multimers as well. So we do have biotin labeled MHC class 1 pentamers, and also obviously the biotin monomers for the class 2 for use for the class 2 tetramers, and you can indeed use DNA barcoded streptavidin in those contexts to do that labeling. We do have a question as well from Andreas. Andreas, if you want to unmute and ask your question.
Yeah, great, thanks. You can imagine that the introductory slide mentioning your class 2 tetramers for detecting potential T cells for other creation got my attention, because one of our concerns, so we didn't go that route yet, because one of our concerns was the frequency. So we would expect as a naive response to be very low. So that was kind of holding us from going that route. But as you put it on your slide, I mean, I guess you can probably share if it was done and then done successfully from other parties, or you have any of this knowledge, or that it was successfully, like, could detect a certain peptide, yeah, from an antibody then in a naive population. I don't know.
Yeah, I mean, these can detect very rare populations. And yeah, we have several of the tetramers against therapeutic antibodies, for example, on our in our catalog. And then we have a nice publication where these were actually used to isolate B cells. So they were even able to isolate some of the very restricted B cells as well. And that's a publication on the website.
Okay, cool. I will check it out.
Yeah.
Thanks.
Yeah. So Andreas,
Anything to add to that, Jeremy?
yeah, just in comment to that. So exactly what you said, Emilee. So I think the epitopes in the catalogue under the therapeutic antibody section are rituximab specific, and they were epitopes that were published by Bernard Meyer, if my memory serves me correctly. And I guess they would typically, I mean, obviously you're absolutely right that the frequencies in these samples can be very, very low in the sort of pre-existing, but these were in RA patients, but actually being able to then monitor post-treatment with rituximab, they were able to then identify those antigen-specific T cells.
Okay, cool. Thanks for the information.
Cool, thank you. It's definitely an under applied area in the context of unwanted immunogenicity. I think there's a lot of work to be done in that area for sure. Good. Well, thank you very much, Emilee. We will move on to our next speaker now in the interests of time. So thank you for presenting that.